Hello, everyone. Thank you all for joining us again for another exciting Tuesday seminar with the International Online Spinal Cord Injury Seminar Series. Today's talk is by our superstar, Dr. Megan Detloff from Drexel University, being moderated by Dr. Bethany Kondiles from my cord. And if you're as excited as I am to be here, please hit the like button down below. And if you haven't already, please subscribe to the iOscars channel. And thank you all for joining us today. And Bethany, take it away.
Well, thank you very much, Sabia. As Sabia said, we're going to have one of our own iOscars superstars today, Dr. Detloff. You might have noticed we had a little bit of a scheduling snafu, so I'd like to thank Dr. Detloff for coming in kind of last minute so we could still have a fantastic seminar today. Because of that, it's going to be a little bit different, maybe than normal, maybe a little bit shorter because she's sort of pinch hitting for us today. So we might not have a question slide right in the middle like we normally do. We might sort of just play that by ear. But regardless, if you have any questions for Dr. Detloff, drop them in the chat. Either preface them with a Q or the @ button so that I will see them. So @ Bethany Kondiles. And without further ado, it's my honor to introduce Dr. Detloff, who did her undergrad at the University of Michigan, Ann Arbor, actually in chemical engineering, which I didn't know until today. And then did her PhD at Ohio State with Michelle Basso, and then moved on to do her postdoc at Drexel with John Houlet. Where she is now actually an assistant professor of the Department of Neurobiology and Anatomy in the Marion Murray Spinal Cord Research Center at Drexel. And you might also know her, as we have mentioned, from her superstardom in terms of everything she does for us here at iOscars. So it's an honor to introduce Dr. Detloff, and I will hand it over to you.
All right. Thank you very much, Bethany, and hello, everyone. Thank you for allowing me to present again. I promise this is a completely different data set than I presented earlier in what I think was our second iOscars Tuesday seminar. So again, I appreciate your continued support. And if you have any nominations for speakers, for people you think would give a good talk, or people you want to hear more about their work, hit that nomination form link that Warren Allalane is hopefully putting into the chat function as we speak. So today we're going to talk a little bit about the role of monocyte-derived macrophages that infiltrate the dorsal root ganglion after spinal cord injury and their association with neuropathic pain. And before I get started, I want to really point out that this work, the lion's share of this work, was done by my first PhD student, Soha Chaya. Yes, her name is spelled correctly. She does have 3 H's in her name, 2 in her last name. And this is a picture of her at her recent online graduation, and then pictures of me in the corner there with a little tear in my eye, because if anybody knows me, they know that I'm a crier. And then there in the bottom right with her and her family. So again, a lot of this is her work, and she's moved on to University of California, San Francisco, and she's a postdoctoral fellow now with Alan Bassbaum. So I also want to take the time to say that in addition to Soha, I think it's really important to congratulate our new PhD and master's trainees who've graduated this year in this uncommon situation, and recognize that we all know that it's hard to get a PhD, and I can't imagine writing my dissertation and trying to defend a dissertation in a Zoom format. So congratulations, good work, and we're looking forward to seeing where you go. All right, now the fun starts. On to the seminar. So I start almost every seminar with definitions, and I want to start by saying that pain is defined by the International Association for the Study of Pain as an unpleasant sensory and emotional experience that's associated with actual or potential tissue damage. I tell my graduate students in class that it's a perception, it's subjective, and it's protective. we require pain as it's evolutionarily conserved because it's protecting us from harm. On the right, you'll see a schematic from the Bible, as neuroscientists, I think, call it, from Kandel and Schwartz, chapter 24, where it shows the ascending pathway for the pain experience. And even in the Bible, in Kandel and Schwartz, it's known to maybe have a few mistakes, and they're missing, in my opinion, one of the most important neurons, the dorsal root ganglion neurons, which are very important for bringing in information from the periphery into the CNS for processing and comprehension. Neuropathic pain is pain that arises from damage to the nervous system in particular. It's described as burning, stabbing, piercing pain. It's intractable, it lasts indefinitely, and it's really terrible. Clinical hallmarks include allodynia and hyperalgesia, and really in the most simplest form, we can consider that as the pain of everyday living. So things that you and I would take for granted, like wearing a t-shirt or shoes, or the air from the ceiling fan here in my office, could be perceived as painful. In spinal cord injury, neuropathic pain is quite common. It occurs in the majority of individuals who suffer or sustain a spinal cord injury. It covers over about a quarter of the body. It can occur at, above, or below the level of the spinal cord injury. The onset can be early, or it can be even almost a decade out, with the majority of onset within 6 months to a year post-injury. What's important, or really not good, is the fact that only 30% of the people who are living with SCI-induced pain, only about a third of them are reporting any kind of relief with the current treatment strategies that are available. And with that, there's only about a 30% reduction in the pain that they are experiencing. So it's really insufficient, and what we've got going on is really, right now, available for these individuals is really not up to what it could be. And so in the lab, for a long time, even, from back in my graduate school days, I really was interested in how neuropathic pain is thought of as a neuroimmune disorder. And so what you can see in this slide, it's relatively busy with the DRG disproportionately large. But what you can see is that out here at the paw, where we would typically test, or within the skin dermatomes, you can see that there's activation of immune cells, and that there are cytokines at the terminal of the dorsal root ganglion neuron that we are most interested in, called the nociceptor, at that peripheral terminal. And then you can see that there's also activation and cytokine, pro-inflammatory and anti-inflammatory cytokines within the dorsal root ganglion, at the spinal cord, and also the brain. So this is something that a systemic, or at least, within the nervous system, there's a close association between the neurons and the immune response that's triggered by either pain, or in our case, a spinal cord injury. And so our question really is spinal cord injury induced pain and nociceptor dysfunction influenced by immune cells? And we've been trying to answer this question in a variety of ways for quite a while now. And so we use a clinically relevant rat model of spinal cord injury induced neuropathic pain. I don't think that I need to introduce it to this audience, but we'll, excuse me, we'll start by showing here on the right is a cadaveric sample post-mortem of a transverse section of human spinal cord. What you can see is white is the spared tissue, the rim of spared tissue, and that the central blackish or purplish part here denotes the hemorrhage, or the area of the core lesion. In the rat, we can model this, and here you see spared tissue, the tissue rim in blue with myelin staining, and you can see the central core lesion. Now, at C5, where we do our contusions, we do them purposefully on one side of the spinal cord so that we can minimize respiratory damage to respiratory centers, because we need our animals to live and we want them to be as healthy as possible. When we do this, we can measure paw hypersensitivity and pain with a wide variety of tests. Today, I'm only going to focus on a classic more segmental response using von Frey monofilaments, where there are different graded plastic monofilaments that are at different diameters, and they bend at predetermined forces. And when we apply these in certain ways to the paw of the animal, we can determine their paw withdrawal threshold. And every rat and just like every person, has a pain threshold, and that's what we're measuring in the paw of these animals. And so, importantly, for any graph that I show you today, you're going to want to know that on the y-axis, less pain is higher, and more pain is lower on this y-axis. And you'll see time on the x-axis. So when we do this in our unilateral contusion, what ends up happening is we see two cohorts of animals. Animals that after a spinal cord injury developed significant paw hypersensitivity and greater than 60% reduction in paw withdrawal thresholds. That's maintained as long as we've looked. We've looked as far out as 13 weeks. And we see that these animals, once they demonstrate paw hypersensitivity, if we do nothing to them, it doesn't recover. Alternatively, we have another subset of animals that maintain normal sensation for the long haul in that ipsilesional forelimb. So on the side of the injury where this lesion is. It's also important, and I think it's important to let you guys know that we also see in our animals, we observe that the same animals that have paw hypersensitivity on their ipsilesional forepaw also show forepaw hypersensitivity. And with this, it's about a 60-40 split, where 40% of our animals show spinal cord injury-induced pain, and 60% maintain that normal sensation. What's important also is that these numbers are from relatively early in my postdoc, and now we're up over 1,000 and close to maybe even 1,500 or 2,000 animals. And every animal that we've tested at chronic time points, we're still falling within this 60-40 split. So this is something that is holding over multiple experiments, multiple experimenters, and students and trainees and myself in the lab. And so we feel like this is really a strong model to use to study spinal cord injury-induced changes associated with pain. We do this, and after we do our pain testing, at about 5 weeks after injury, what Soha did was she dissected out DRGs, and here you'll see staining for ED1 or CD68, staining in brown, visualized with DAB, and counterstained with crescent violet, so you can see the nuclei within the DRG. And what you can see, I hope and appreciate, is that in the animals with injury and pain, so this is again at 5 weeks post-injury, you can see that there is an increase in ED1 positive cells within the dorsal ganglions associated with paw dermatomes. And she quantified that, and indeed we see that that spinal cord injury, there's a slight elevation compared to normal. That dashed line here is indicative of a naive number, or the resident macrophage population, and you see a robust increase there that's significant in the animals with pain. This also correlates when we go, and she correlates this with their pain behavior. We see a nice correlation with the pain score. So macrophage number is correlative with pain score. So higher number of macrophages, you're going to be more sensitive. So our question became, at this chronic time point, are macrophages involved in the persistence of pain, right? Are DRG macrophages involved in the persistence of pain? And what you can, excuse me, see here is an experimental design where we're going to do our unilateral C5 contusion. We know that macrophages infiltrate the dorsal root ganglion. Around 7 days, they persist. And that corresponds with pain. But instead, what we're going to do is we're going to block that. We're going to use liposomal clodronate. We're going to inject it intraganglionically, and we're going to deplete the macrophages once pain is established, and we're going to measure pain behavior. And this is what we found. So again, remember that more pain is down in the lower quadrant. And in animals with pain, what we see is that clodronate and depletion of the macrophage had no change on paw hypersensitivity. And this suggests to us that macrophages, the macrophage presence in the DRG isn't involved in the persistence of pain. Now, because of our model, our injury model, where we have animals that don't develop pain, when we look at those animals and administer liposomal clodronate, we see a dramatic reduction in the paw withdrawal threshold. Every single animal, and this is an N of 9 per group. I apologize, I don't have the individual data points here for you. But every single animal in this no pain group, when we depleted the macrophages, their paw withdrawal thresholds went down. And so to us, that implicated to us that these macrophages might be involved in maintaining normal nociceptor activity, and they could be preventing nociceptor excitability and preventing this pain behavior that we see.
Okay, hey Megan,
yeah,
we might interrupt you. We have a couple of questions
sure.
in the chat, so we can just do a little more informal than we might normally do.
Okay.
Nikos Kiritsis has questions about the impact force of the injury.
Oh, sure.
He had a follow-up question about whether or not the hypersensitivity of the paw is dependent on the severity of the injury.
So Nikos, that's a great question. So the first one, this is the easy part. It's a moderate contusion. It's at 200 kilodynes of force. We do use an impactor probe that's smaller than the standard probe that's there. I don't have the number on the top of my head right now. I'm blanking on that, but I can get that to you if you want it. The other thing is, in terms of this cervical model, we haven't tested it. And I haven't done different severities of contusions at C5. We have done different severities when I was in graduate school in Michelle's lab at Ohio State, where we did graded contusions using the Ohio State injury device, contusion device, and we saw that it certainly was an injury-dependent, severity-dependent condition. So the behavior wasn't there unless we saw at least, I think it was at least 60% axonal or white matter loss at the injury epicenter, but that was at a T8 with a T8 contusion. And the incidence or the number of animals that developed SCI-induced pain in that thoracic contusion model increased as injury severity increased. We also had confounds, I should also say, that as you increase the severity of the contusion, it becomes more and more difficult to test, at least with von Frey monofilaments, it becomes more and more difficult to test because the animals don't weight bear on their paws. And so it ends up being a tough, you end up with more proprioceptive input as opposed to tactile only. So there ends up being some behavioral testing confounds or caveats there that make the incidence a little bit harder to determine.
Okay, we have so many questions flying through. Thank you, everyone in the audience. This is awesome. I'm going to just do a couple more, and then we'll let Dr. Detloff go on. So Stephen Crone has a question. do you see the macrophages in both the ipsilateral and contralateral DRGs, or only ipsilateral?
So that's a great question. I'm surprised this isn't an interneuron question, but thanks for your question. Yes, the answer is yes. We do see macrophages showing up in the dorsal ganglion on the contralateral side. I haven't quantified that, but I have preliminary data that shows that. And when we start probing for different cytokines and chemokines, we also see that there are changes there. But again, that's relatively preliminary, and I don't have as high of an N on that contralateral side yet.
Okay.
But thanks for the question.
We'll just do one follow-up that you're kind of hinting at already, but Inez Maldonado was wondering about the macrophage phenotype that you look in the DRG, or any potentially levels of cytokines, as you were just alluding to, with and without them. Yeah,
so stay tuned. That's
so if you speak briefly to
yeah, keep listening. We'll get to talking a little bit about that, and that's certainly where we're going. I didn't include it in this talk, but we have information that Soha collected as part of her dissertation work, where we did a time course experiment, and we looked at early time points post-injury, really acute time points at 12, 24, I think 48, 72, and 120 hours post-injury, or 5 days post-injury. And we looked and watched the macrophage phenotype sort of change over that early time period. And so that's a paper that's in revision right now in Scientific Reports, and I'm hoping to get that back out for re-review here shortly. So look for that in the coming weeks. But the short answer is there are differences. In those early times, we see a lot of variability. Now that's also, we can't correlate that to pain, because that precedes when we can actually test for pain in our injury model. But there's a lot of evidence related to that, and I'll get to some more cytokine and phenotype markers at more chronic time points later in the talk.
Okay, fantastic. Well, then why don't we let you get back to it? And everyone keep those questions coming. I see them, and we'll try and hit them up again in a little bit.
Okay, sounds good. All right, so we know that the macrophages, so from the quadrinate experiments, we know that the macrophages don't appear to be involved in the persistence of pain, and we have some evidence here where they might actually be playing a role to maintain nociceptor activity and to sort of maintain normal sensation. And so our question then shifted to looking at the DRG macrophages involvement in pain development. And so again, we're doing the same unilateral C5 contusion. We're looking at, we know that pain, we can accurately and reliably test for pain at 7 and 14 days. We can get a pain phenotype there, and we know that our macrophages are coming in early. And we're taking advantage of the fact that there's a monocyte chemoattractant protein, CCL2, which is responsible for pulling bone marrow derived macrophages out of the bone marrow and into the blood. And so CCL2, I like to tell people who don't really understand or know about immunology, and in particular my parents, we talk about this all the time, that CCL2 is kind of like a bat signal, and that it comes on and it's shined in the sky, and then Batman appears. And so the monocytes are kind of Batman in this scenario, and CCL2 is a bat signal. So we're taking advantage of this naturally occurring thing, and we're going to manipulate the macrophages by manipulating CCL2 and its interaction with the receptor that's found on monocytes. And so in order to do that, we had to run an ELISA and make sure that CCL2 was indeed present in the dorsal root ganglion after a spinal cord injury. We've done that. Soha did that, and we can see that it's a short and transient bat signal, and it's there and significantly upregulated. Excuse me, sorry about that. Within the first 24 hours, it comes back down to normal by 3 days post-injury, and it stays at at normal or below normal levels, even when you go out to 1 month post-injury, at every time we've looked. We've also done corresponding experiments using qPCR to probe for the gene expression of CCL2 and whole DRG, and we see very similar results in that gene expression in the DRGs. It's also important to know, and I'll add here, this is for the ipsilesional, that ipsilateral DRG. We also see evidence of CCL2 upregulation in the contralateral cervical DRGs and also in the bilateral lumbar DRGs as well. So those DRGs that correspond to the dermatomes of the paw, we're seeing CCL2 upregulation in this short transient fashion. And we know that just following CCL2 upregulation, we wanted to make sure that macrophages or ED1 positive cells show up within a reasonable time frame, and we do see that as well. So we're fairly confident that CCL2 can be the bat signal that's bringing these cells in. And we're currently doing experiments now with lysim EGFP mice to determine whether or not, to further understand whether or not these are cells infiltrating into the dorsal root ganglion after an injury, or if it's a question of proliferation. But in the rat, we're not capable, at least right now, at Drexel, to do bone marrow chimeras to ask those types of questions. And so when we use CCL2, what we're doing is we're actually going to use an inhibitor. We're going to give it intravenously. We're going to inhibit the CCL2 receptor called CCR2. That's, here is represented here in the schematic with the macrophage with the CCR2 receptor. And now the monocyte or macrophage won't be able to infiltrate the dorsal root ganglion because it's not going to be able to perceive the CCL2. Importantly, we're only giving that in the early time period when we first see the monocyte-derived macrophages come into the dorsal root ganglia. And when we do that and we measure pain, so what you can see here is, again, a von Frey monofilament test, where on the y-axis is the paw withdrawal threshold of the animal, and along the x-axis is time. And you'll have our 3 groups here. And so you want to pay attention to the red group of animals. So when we test 7 days after the spinal cord injury, we were incredibly surprised. As David Magnuson says, if I'd bet the farm, I'd have lost. What you can see in our vehicle-treated animals, we're still at about that 60-40 split, where in these experiments with Ns of over 9, we have 33% of the vehicle-treated animals showing pain-like behavior, and about 2/3 of them exhibiting normal paw withdrawal thresholds. When we treat with INCB and we prevent macrophage infiltration into the dorsal root ganglion, we were surprised to find that nearly all of the animals developed paw hypersensitivity within that first week. Now remember, the drug is only on board for the first 3 days after the spinal cord injury. When Soha went and tested at subsequent weeks post-injury, we see that the incidence of pain is in the INCB treated animals is returning more towards that 60-40 split. And so what that's really suggesting to us is that the macrophage presence is important, again, for maintaining normal sensation in the early time points after spinal cord injury within the DRG. So when we do that, and this comes to the question that was asked at the break, is that when we block macrophages from infiltrating into the dorsal root ganglion early, this is kind of what we see. So this is PCR, quantitative PCR of whole DRG tissue on the side of the injury. So on the same side as I'm showing you pain behavior. And the black bars that are naive, blue is vehicle, red is our drug-treated group, which should have fewer macrophages or monocyte-derived macrophages within the dorsal root ganglion. And you're seeing in markers of pro-inflammatory or more M1 markers of macrophage phenotype, iNOS, and CD80, you're seeing that there's a significant reduction compared to naive and vehicle both at 7 days after the injury, and that more pro-reparative markers, or M2 markers, as some people like to call them, arginase 1 and CD206, are low in both groups. So that's suggesting that the early monocyte-derived macrophage or macrophage response in the DRG is more of a pro-inflammatory nature. We've tested other phenotype markers that I didn't put on this slide, and they're really falling within these same trends. When we look at these same things at 28 days post-injury, we see that, excuse me, there's a shift in the animals. You can also see this large degree of variability within each group within each experimental group, and that there's this elevation, in particular in iNOS, compared to the naive or uninjured animals, but there's no difference between the vehicle-treated or the INCB-treated animals at this 28-day time point. And when we look at the other markers, we see that both vehicle and INCB-treated animals are moving in similar directions. So they're very similar at that 28-day time point. And to us, this suggests maybe one of two things. One is that macrophage infiltration is delayed because of the INCB treatment, because we're manipulating that CCL2, CCR2 signaling, BAT signal, or that the macrophage presence in the chronic DRG or at 4 weeks post-injury is due to the proliferation of resident macrophages, of the resident cells. When we look at the cytokine profiles within the DRG, and again, this is quantitative PCR, looking at whole DRG on the side of the injury. We also have contralateral, and I'm not sharing that with you today. But what we're seeing when we look at certain pro-inflammatory cytokines, we're seeing that at 7 days, when we saw our behavioral effect, that TNF-alpha is low compared to vehicle-treated animals and to the naive controls, and that INCB-treated animals also have less IL-6 and IL-12 at that 7-day time point. And not surprisingly, IL-10, which is an anti-inflammatory cytokine, we're seeing a reduction there, but that sort of goes along with the fact that we're not at the 7-day time point seeing many M2 or pro-reparative markers of macrophages. When we look at 28 days, again, we're seeing, and I'll just pull all of these up, we're seeing that, again, both injury groups, both treatment groups, the vehicle and the INCB group at that 28 days post-injury are acting similarly. You see a reduction again in TNF-alpha, in the gene expression of TNF-alpha, an increase in IL-6, and IL-12 is also reduced in those populations compared to naive animals. And within the M2 or anti-inflammatory cytokines, what we're seeing is a significant reduction in INCB versus naive only. And so what that suggests to us is that macrophages are a source of DRG cytokines post-injury, because we're seeing that when we deplete them, there is a reduction in cytokines, both good, maybe good, and bad cytokines, for simplicity there, and that when we take the break off of the macrophage and we allow them to repopulate or come back into the dorsal root ganglion, it's now acting like a spinal cord injured animal that was treated with vehicle. And so this is what I've told you happened in the DRG after a spinal cord injury. So here is my attempt at an animated schematic. So here we go. So on the right here, you can see my drawing of a nociceptor. The first thing that we do is we see CCL2 release in the dorsal root ganglion. There is evidence that it occurs, it's secreted by neurons. I told you that I think that the CCR2 positive macrophages are recruited by that bat signal CCL2, that the macrophage differentiate. The monocyte-derived macrophages are going to differentiate. They're going to release cytokines, more good than bad, and that those cytokines are going to alter ion channel activity within the nociceptor. And that's going to then be associated with the presence or absence of pain behavior. So what I wanted to do, and what we've also been trying to do for a long time with the help of a very important mentor of mine, Terry Walters, in Houston at University of Texas Health Science Center there in Houston, is to be able to record from these cells and to get some information about the excitability of nociceptors in situations where we manipulate the macrophage response after a spinal cord injury. And so this is preliminary data. I'm sharing it because I think it's important that we really think about how the immune system or the monocyte or the immune cell or whatever cell that's non-neuronal is interacting with the neuron and how it's bidirectional. So here's our preliminary data. So we started doing whole cell patch recording of dorsal root ganglion neurons that are presumptively nociceptors because they're very small. They're IB4 positive. And so we're recording, we're dissecting these neurons, plating them in tissue culture, and we're recording from small cells when we apply different amounts of current. And so what you can see in this trace is a cell, 3 different traces from the same cell. The first, where we give a hyperpolarizing current, which is going to inactivate the cell, and that's indeed what happens. And then as we apply more and more current, you see that it fires an action potential over the current step, a single action potential. And if we increase the current even more that we inject into this cell, it's able to fire multiple action potentials. And that's an indicator that you are indeed firing, you are indeed recording from a nociceptor. And so those were all good things. And that was a huge win for me. And I was very excited when I saw this data a long time ago. Now, the electrophysiologists know that we can take this data, these traces, and we can measure any number of membrane properties and get information about the stability or the excitability of these cells, how likely they are to fire action potentials, and things like that. And so some of these things are things as simple as the resting membrane potential, where you just connect to the cell and you don't do anything to it, and you just find out what its membrane potential is. The membrane resistance, which is kind of for simple terms, is this sort of a measurement of the ease of ion flux across the membrane, and then rheobase, which is the amount of current you have to inject to get that first action potential. And what I can tell you with 3 animals per group right now, where we did the same experiments as before with the INCB, and we're recording at 7 days. So when we would see the behavior, we're not seeing any differences. And I don't think in nociceptor, in membrane potentials, or in indicators of nociceptor excitability. However, I think the jury's still out here. These are some of our first recordings, and we need to certainly follow that up because we've learned a lot now. We've learned a lot more now about whole cell patch experiments and how to do them and things like that, in large part with our collaboration with Terry Walters again. And so again, this is that same slide where I said, this is what I told you, but this is what I left out. I left out the fact that CCL2 is actually pleiotropic. It has multiple functions in the body, and one is that CCL2 can actually act on neurons themselves. And so So we know that some DRG neurons have CCR2 receptors, and that activation of this receptor can turn on intracellular signaling pathways that can modulate cytokine receptors on their expression levels, and also whether or not they're found on the membrane. And it can also alter ion channel insertion into the membrane and ion channel gene expression. So this is certainly something that we are keeping in mind, and we are moving forward to try to understand better, and really try to understand how these chemokines can impact neurons directly, as well as indirectly via the immune cells. And so, to summarize what I've told you about today, is that we've really found nothing yet to indicate that the macrophages that are in the DRG after a spinal cord injury are actually bad. I've shown you evidence that there's a temporal and evolving role of the macrophage in pain development and maintenance after the injury, and that the macrophage can mediate normal sensory phenotypes after spinal cord injury, and it might actually be in terms a lot of us like to think about as being potentially neuroprotective. And thirdly, their elevated presence in the dorsal root ganglia after injury might be an indicator of neuronal dysfunction. We do know that, and Terry has shown several years ago now, that nociceptors are hyperexcited after a spinal cord injury, and that they are more spontaneously active. And so we really want to understand whether or not, what the contribution is of the macrophage on the stability of those cells, and we're working towards that. And the way that we're starting to think about this is that there's really a conversation between the macrophage and the nociceptor. And I think one of the buzzwords that we hear all the time is this idea of neuroimmune interactions, and it just seems like this sort of catch-all word that doesn't really, it didn't really speak to me for a long time. And so once I started thinking about this potential dialogue between these two cell types, and that it's bidirectional, it really opened up a lot of ideas for me and thoughts about how to move this field forward. And so this is something that, in thinking and bringing it back sort of to a therapeutic, a little bit more of a therapeutic perspective, if we can manipulate a pro-inflammatory macrophage in the DRG, or the pro-inflammatory response in the dorsal root ganglia in that sort of subacute time period, we might be able to prevent aberrant pain behavior. And this can be really attractive because the dorsal root ganglion can be an attractive target because it is not within the CNS. You don't have to try to cross the blood-brain barrier. You can also selectively target individual DRGs if it's a very localized pain dermatome. And it just provides a potential avenue that might be a little bit different. And so with that, I would like to acknowledge the people in my lab who have done the work. Again, it's Soha Chaya. The lion's share of the work was Soha Chaya. Or LJ Richards in the lab, he was really helpful in doing some of the clodronate experiments in the surgical procedures there. Packlink, Theodej Thawira-Tannison is a patch electrophysiologist, a former postdoc in the lab, and he helped get our patch experiments up and running. And my longtime techs, Amy Ong and Daniel Quiros Molina, who are responsible and helping with a lot of animal care. I'd be remiss if I didn't acknowledge my other pain pals, Terry Walters, Alexei Bavenkoff, and Ona Bloom, as well as the Marion Murray Spinal Cord Research Center, our incredible group there that was headed by John Houlet for 15 years and has recently, the reins have been handed over to Simon Gister. So I couldn't do it without John's support as a postdoc and his continued mentorship as I grow up in the academic world. And Kim Doherty has been my go-to person and on-site help for patch experiments, and of course the financial support from the NIH and the Craig Nielsen Foundation. So I'll take any questions that you have. Thank you.
Great, thank you so much. Thank you for making it hard to be a moderator, that I was paying so much attention to the talk. It was hard to also pay attention to the questions. So we had quite a few people asking some similar questions, which I'm going to try and roll together if I might.
Okay.
Sort of just asking, maybe we can just, if you just want to, wax poetic for a bit about what these treatments do, what your treatments do, or what you kind of see with these sorts of macrophages a little more globally. So whether it be in terms of the injury sites specifically, or I think Morgan Sharp, let's give one of our trainees a shout out. Sharp asked specifically about macrophages in the DRGs above and below the injury itself. And then I think we also had a question about infiltration into the entry zone and that sort of thing.
Sure.
So it's kind of a big question, but if you just kind of wax poetic about effects throughout the whole of the cord, I guess.
Okay, I can do that. So first of all, Morgan, I owe you an email, but the short answer is yes. And then to answer your question here, we haven't probed beyond sort of preliminarily rostral or caudal DRGs. What I can tell you is that we're predominantly looking, and all this data was looking at C7 and C8 DRGs on the side of the spinal cord injury. But we have also looked in lumbar DRGs on both sides and the contralateral side. And what I can tell you is we are seeing evidence that falls along the same lines, where we're seeing increased macrophage number at chronic time points post-injury that correlates with pain behavior of the corresponding paw. What I can, and so that's about where we're at in terms of looking for cytokines and chemokines for the most part. We have only very preliminary data, ends of 3 there. So because of the variability in the normal animal, I'm in the DRGs, I'm less confident in giving you a concrete answer about, in the lumbar cord 20 segments below our lesion, or in the lumbar DRG at this point. In terms of what happens at the injury epicenter, this is a great question, and this is one that I talked with Mike Beatty about as I was even before Soha was probably even in the lab. We had talked for a while about this sort of, if you manipulate the dorsal ganglion macrophages and if you do some of these systemic approaches, how are you not manipulating what happens at the injury epicenter? And the data that I neglected to show you today is the sparing data. So we do contusions when you do incomplete injuries. I think it's important to know what your injury severity really is beyond just the number you get on the IH device. And so we do all that, and we look at spared tissue volume and lesion size, and we're not seeing any significant differences when we do that. And further, when Soha looked at least at 28 or 35 days post-injury, we don't see a difference in the degree of ED1 positive cells or even IBA positive microglia and macrophages at the injury epicenter, either at the section that would be considered the epicenter or at the rostral and caudal poles of the lesion core. So, what's important is the 2 strategies that we used here, which I don't think in any way are necessarily therapeutic, but more to get at a mechanistic, answer some mechanistic questions. We're not seeming, because of the strategies we used, and one of the reasons we only wanted to apply or give INCB for 3 days was that we didn't want to mess up the normal or immune and inflammatory response that occurs at the site of the spinal cord injury if we could help it. And so we started with claudronate experiments, mainly because we could localize the macrophage or myeloid cell depletion to specific dorsal root ganglia and have less of a systemic effect. And then the INCB we only gave for those 3 days. So we're really aware that we could be changing things at the injury epicenter, and we're trying hard not to do that. The other thing that I want to say is that when you look at pain centers within the dorsal horn of the spinal cord, when you look at pain centers in the dorsal horn, you can see that there is a pain-specific increase in microglial activation. Again, I showed that as a graduate student. Brian Haynes and Stephen Waxman, when he was a postdoc with Stephen Waxman, also showed that after a mid-thoracic contusion. And we've recapitulated that in the cervical contusion also. What's cool is that, and this is published in the Journal of Neurotrauma paper that Soha was first author on, what's really cool is that when you actually exercise animals, I have a pretty strong experimental evidence that early exercise intervention with a wheel walking exercise can prevent paw hypersensitivity and prevent pain, and in the majority of animals. And one thing that we can do is we can actually take, when they're just a fraction of those animals, very small amount, about 10% of those animals are refractive to this wheel walking exercise. And when Soha looked at those animals, the macrophage numbers in the dorsal root ganglion were still high. But when she looked and stained with IBA1 in the dorsal horns of the spinal cord, she saw that the wheel walking exercise actually reduced IBA1 positive staining, but it didn't change the peripheral macrophage number. And so what we really think is happening is that it's really these peripheral macrophages that are more important for the presence or absence of pain. So in animals where exercise, they were resistant and they still developed pain despite this analgesic exercise paradigm, they still had high macrophages in the DRG. There's also a lot of evidence, and then I'll stop waxing poetic so you can ask another question. There's also evidence that there's supraspinal activation of sensory and pain pathway nuclei in the brain. So in places like the thalamus and the S1 cortex, there's a lot of information about the electrophysiology, and there's information that there are macrophage and microglial activation, at least, within the thalamus, S1 cortex, anterior cingulate cortex, those sort of pain centers there. And that neuroimmune hypothesis and glial hypothesis of pain can also be extended to comorbid depression, where there's a lot of data that suggests if you can treat systemic inflammation, you can also reduce some major depressive disorder. And so there's a lot of comorbidity between pain and major depressive disorder. And there's also commonalities within brain regions and neural circuitry that underlies both of those conditions. And there are neuroimmune hypotheses associated with both of those. So those are just sort of my tidbits, I guess, related to what's going on with the monocyte-derived macrophages or microglia after a spinal cord injury, close and far away from the lesion.
Yeah, I threw you quite the big question, so I appreciate that. It was super interesting. I want to then just hop to a question that Hadi A had a couple of questions actually, citing some work in nerve injury.
Yeah.
Looking at the role of I guess someone, he said it was something the name of Basbaum.
Yeah.
So looking at CXCR1 instead of CCL2.
Yeah.
But just because you were just talking about sort of this larger mechanistic, the supraspinal, and then what's also going on in the dorsal horn. And now we're going to look at maybe what might be going on peripherally. If you could kind of comment on how that all kind of comes together and what the compare and contrast peripheral versus central injury.
Yeah, sure. So there are a lot of similarities between peripheral nerve injury models and spinal cord injury models of pain. Certainly when we look at things like nociceptor excitability, nociceptor primary afferent sprouting into the spinal cord, certainly the glial hypothesis of pain, it all came from the peripheral nerve injury world. And Alan Basbaum in his lab, and that's actually where Soha is now, but she's not working on this project, has recently published a paper in Nature Communications, I believe, where they looked at CSF1, which is the colony stimulating factor, and the proliferation of macrophages, and how that is associated with pain development and things like that, looking in the dorsal ganglion and also in the spinal cord. And they actually see things that are a bit different. And it's my understanding and some of the thoughts that I have about why what we see and what they see is different is, one, that they use a variety of different animal models, but the injury is happening to the primary nociceptor. And so the injury is happening because they're manipulating the peripheral axon of the primary sensory neuron, the one that's out closer to the skin, as opposed to what we're doing. We're not actually damaging these nociceptors directly at all. It's an indirect activation or an indirect cue that's going to cause this nociceptor to change its gene expression or its responses. So the spinal cord injury, if anything, it's damaging the large diameter DRG neurons that come into the spinal cord and ascend up in the dorsal columns, and it's going to have very little impact on nociceptive primary afferent axons that are coming into the dorsal horn, just because of the location of the lesion relative to the C7 and C8 DRGs and those that are associated with the dermatome of the paw. So I don't know that that's the best answer, but I am aware that paper is out there. I've been thinking about it a lot. It's very different than what we see, and I'm surprised by that. And we're working to try to understand and better understand, really what that means and what the similarities and differences are in the DRG. I'd also like to point out that Brad Kerr, who is also a pain guy, he trained with Sam David, and he's a neuroimmunologist, also looks in the dorsal root ganglion, but instead of spinal cord injury, he looks at he uses MS as a model of pain. And when he does that, he's also noticing that there are drastic immune changes in the dorsal root ganglion associated with MS that affect pain behavior, and that exercise can also modulate the pain development in these EAE type models of pain. Next question.
That's fantastic. I'm learning so much. I think I'm going to be a good moderator, though, and we'll just do one more quick mechanistic question, and then there are so many more questions. There's a conversation going on that I totally lost track of, so you should definitely go check out the chat.
All right.
Here. But I saw it a couple times. Some people were asking about genders of your or sexes of your animal.
Yeah, that's a great question. So the quick answer is we've only done this in females. We have to replicate and validate this in that similar things are happening in males. And again, we have a mouse colony that's up and running, and so we're moving in that direction now.
Okay. Well, with that, I'm sorry, I see so many fantastic questions in the chat, and we don't have time to get to all of them. Okay. So thank you very much, Dr. Detloff, for that fantastic talk. So with that, I think we'll end up today's session. And so join us again next week. Dr. David Magnuson from the University of Louisville is going to be presenting The Bad and the Ugly of Rat Physical Therapy After SCI. What? No good? So maybe a little continuation from what Dr. Detloff was presenting today about exercise and its effects on pain. Maybe, come find out. It'll be moderated by Dr. Amalia Dov from Duke University. So again, thank you very much, everyone, for joining us. Thank you very much to Dr. Detloff, again, for pinch hitting, coming in today with a scheduling snafu. Hang out in the chat, and we will see you all next week.